Okay, well, good afternoon or good evening, everybody. I guess it depends where in the world you're listening to this webinar from. It's a great pleasure to be here this evening. As Kat very kindly said, I'm going to be discussing using non-animal models to study breast cancer. So I'm based at the University of Aberdeen. You might be wondering where Aberdeen is. Well, we're right up here in the northeast of Scotland. To the south, about 2.5 hours is Edinburgh, and further south, about 8 hours away, is London. So we're quite far north in the UK. The University of Aberdeen, as you can see, was established in 1495, so it's one of the ancient Scottish universities, but we maintain some of the ancient traditions alongside more modern architecture around our two campuses. I'm based at the Forester Hill Health Campus, which means that we have access to patient-derived samples, and we are allowed to interact. We have the privilege actually of interacting with clinicians on a daily basis. So that can really help speed up our research. And I've been lucky that for most of my career, I've been based in hospital sites, and actually it's been easier to use human tissues in research than it has been to use animal models. And as Kat said in the introduction, that's been very much a focus of my research over the years. So I'd like to start off by showing this timeline of breast cancer models. And as you might expect, the first transplantable mouse mammary tumoral model was actually in a mouse, and that was just at the beginning of the century before last, 1903. But it wasn't until 1958 that the first breast cancer cell line was developed. And that's the BT20 cell line, which some of you may have used during the course of your research. In 1973, what I would consider the most commonly used human breast cancer cell line, and every breast cancer lab that does cell culture will have these in their stocks. That's the MCF7 cell line, and that was derived from a pleural effusion from a patient who had estrogen receptor positive breast cancer, and it's exquisitely sensitive to estrogen and has been used as a model to understand how estrogen and anti-estrogens work towards combating breast cancer. In 2003, there was a seminal publication, which I'll describe in a little bit more detail in a moment, an editorial published in Nature. And also at that time, the development of patient-derived xenograft models were described. In 2015, so about 7 years ago now, so-called patient-derived organoids were described, and these are sort of a step up from the patient-derived xenografts, which use animals. The patient-derived organoids use only human tissues. And in 2017, the Breast Cancer Now Tissue Bank has published work using fully humanized models to study early breast cancer, and that's using tissues and cells which have been derived from the Breast Cancer Now Tissue Bank, which Kat mentioned in her introduction, and I'll say a little bit more about as we go through this webinar. So that's the timeline of the various breast cancer models that have been used over the years. In terms of current models, there are 4 different model types that people are starting to use, and I'll talk through all of these, except, of course, the PDX models, because, of course, this webinar is about non-animal modelling. So I will discuss 2D cell culture, I'll discuss organoids and tissue slice models, and I'll also touch on some of the microfluidic models. As well as some other things at the end, which I hope you'll find interesting. So, for many years, people have used cell lines to study breast cancer, not just breast cancer, of course, but other types of cancer. And the beauty about cell lines is that they grow very easily. They grow in 2 dimensions in tissue culture flasks, similar to the ones shown here. and they grow on the plastic substrate, and they form this confluent monolayer. And they've been used for, as I say, a number of years. The first immortalized human cell line was the HeLa cell line, which was obtained in 1951 from cells taken from Henrietta Lacks during treatment for her cervical cancer. And they were instigated in Johns Hopkins laboratory by researcher Dr. George Gay. However, breast cancer cell lines, and indeed all cancer cell lines, are a rather simplistic model because they just comprise the cancer epithelial cells themselves. And of course, we know from looking at human breast cancers down the microscope, or more typically these days on a digital image, such as the one shown on this slide, that breast cancer is a very complex and heterogeneous disease. And the cancer cells shown here do not exist in isolation, but rather they coexist amongst a complex architecture of different cell types. For example, the stroma, and within the collagenous stroma, you will find things like carcinoma-associated fibroblasts, so these long, slender cells. And we know from various studies that originally these cells were thought to be supportive. Originally these cells were thought to not really have any function, but we now know that they can send bidirectional communication to the cancer cells and influence the behavior of the cancer cells. There are also adipocytes, and increasingly we're learning that the adipocytes as well can send signals to the tumour cells and instruct them to do certain things. Often within breast cancers, there are immune infiltrates, so these are the small darkly stained cells shown here. So tumour cells do not exist in isolation, but rather against a very complex backdrop of various different cell types, which can all help send signals to communicate to the tumour cells and give them instructions, tell them to do various things. While I was preparing for this webinar, I took a look at PubMed towards the end of last week. And I mentioned about the simplicity of two-dimensional cell culture. But more recently, three-dimensional cell culture has been very popular. And I looked back at PubMed from 1980 through to 2020. And you can see for the first roughly 20 years, there wasn't an awful lot of difference between the number of studies using cell lines versus the number of studies using 3D models. And I remember these days, I started my scientific career around about this point in the 1990s. But then, as you can see, in 2003, something started to change. We're starting to see a bit of a separation in the number of models using 3D cultures, the red line, whereas the number of publications using 2D models was starting to slow down a little bit. And that, I believe, was a result of this editorial, which was published in Nature in 2003, so 20 years ago now, and it was called Goodbye Flat Biology. And that editorial was really encouraging scientists that they should be moving away from growing cells in isolation in flat tissue culture plastic. So moving away from this reductionist approach to aim to identify more complex models which better recapitulate how cancer exists when it's in the body. And in, I think it was around 2016, the first conference called Goodbye Flat Biology was held. That was in Berlin, and I was fortunate enough to attend that conference. And I believe now that the fifth incarnation of Goodbye Flat Biology is taking place in October of this year. And I'll give you links to that at the end of the webinar. They're really excellent conferences if you want to get into modelling cancer in 3D. So I introduced you to the rather simplistic a 2D cell culture model. And of course, the next step up from that is to try to generate 3D cell cultures. And you can do that from traditional cell lines. It's not that difficult to do. And typically, the cells will grow in these 3D spheroids. And in the breast cancer field, very often they are described as mammospheres. And you can encourage the cells to develop these 3D cultures either by putting a coating of agarose on the base of the cell culture dishes, and that prevents the cells from attaching, and they're forced to form these 3D structures, these 3D spheroids. Low attachment plastic will do exactly the same. And you can also use the hanging drop method, whereby small numbers of cells are forced to grow on the lid of a culture dish in the form of this hanging drop. And again, the cells are forced to coalesce and form these 3D cell cultures. People have also looked at different matrices as a way of encouraging cells to grow in 3D. The first one of these is Matrigel, but one of the downsides of Matrigel as we move towards moving away from animal components in cell culture is that Matrigel is actually derived from animal, it contains animal components. People have also used collagen, and if you want to move away from using animal components, you have to use human collagen, and that can be quite expensive in comparison to the rat tail collagen that has been used traditionally in the past. And there are also these peptide hydrogels, which have become available much more recently. And these hydrogels, if you like, they're a blank slate. They're a basic gel, and you can add different things to the gel to functionalize them to make them more comparable to the substrates that cells would find when they are within a tumor. And we've done some work using the peptide hydrogels. They're quite difficult things to work with, but if you persevere, you can get some very nice data. And a great advocate of the peptide hydrogels who have done some really nice studies are Professor Kathy Merry at the University of Nottingham in the UK. And her postdoc, Jenny, they have done some really very nice studies using peptide hydrogels and growing cells in 3D in these. And I would encourage you to look at their papers if you want to find out more about these models. But of course, when you grow cells within a matrix, sometimes they can be difficult to remove from the matrix if you want to then do downstream studies. So some years ago now, we came across this culture media called Epifiber Life. And as the name might suggest to you, Epifiber Life was developed to support the growth of fibroblasts. And indeed, it does support the growth of fibroblasts very nicely. The fibroblasts are a lot more robust in the fibroblast media than they would be in a standard culture media. But in order to make the Epifiber Life, you have to add a few components which come with the Epifiber Life in the form of a bullet kit. And we thought, well, actually, what happens if we modify this Epifiber Life by omitting some of these components? And we did this, and we called it EpiFL, so Epifiber Life. So with this Epifiber Life, rather remarkably, what happens is that when you gradually introduce the cells to Epifiber Life, the cells start off as a conventional monolayer, plastic monolayer, over time. And this process happens fairly quickly with a gradual introduction right up to 100% Epifiber Life. The cells start to grow in these 3D clusters, so quite naturally within the cell culture flasks, in the absence of any media, any matrix support. And we can see this with different types of cell lines. And this is, here we are here, the two different cell lines. So this is MCF7 cells, which form a tighter cluster, but some of the non-luminal cell lines form these much looser structures, much more reminiscent to like bunches of grapes. And here we have a representation of various different cell lines growing in phase contrast in EpiFL. And we've taken sections of these spheroids. Here you can see H and E. We've stained with Ki-67. And typically what you see is an outer layer of proliferating cells, which is stained with Ki-67, and apoptotic cells in the center when you stain with an apoptosis marker. The cells which form very nice, tight, adherent clusters stain positively for E-cadherin, which is probably not too surprising. The three here, which are forming the nice, tight clusters. But where you see the more grape-like structures, you don't see the expression of E-cadherin. So the Fiber Life can be modified to form EpiFL, and that supports the spontaneous growth of breast cancer epithelial cells in 3D culture. And we've gone on to do some co-culture experiments with the carcinoma-associated fibroblasts, or CAFs, that I mentioned earlier. These are the cells that can support the growth of cancer epithelial cells. And our CAF cells were labelled with a red marker, as shown here. So what we did, we allowed the cells to develop the spheroids first of all. We then added the CAFs, and the CAFs started to stick to the outside of the spheroids, and after 96 hours they had started to invade into the centre of the spheroids. And this is shown in an immunohistochemical image of the sections which have been stained, sections of the spheroids. So here you see the cancer cells stained with Ki-67 on the outside, and the fibroblasts. The fibroblasts had a marker, an RFP marker, and you can see the cells on the outside of the spheroid, and over time they start to infiltrate into the centre of the spheroid. And it's a little bit difficult to see, but this is a tissue microarray core here of a breast tumour, and the way that the fibroblasts are arranged within the spheroid are similar to what you see within a breast cancer. So a nice model if you want to look at the interaction between CAFs and cancer epithelial cells. And this work has all been published previously. So back to our models. So you may be aware that in the US, the NCI has this NCI 60 cancer cell line screen, and they use this cell line screen as a way of identifying suitable candidates to take forward as potential new drug therapies. But in 2016, there was an article published in Nature which stated that these cells were to be retired and they were actually going to be replaced by human tumours grown in mice. So that was a little bit surprising to me, and this was an image from the paper. And they wanted to optimize mouse models for precision cancer prevention. And like I said, that was a little bit of a surprise to me. Why would you want to use mouse models as a precision medicine approach? Because, of course, we know that a mouse is not equivalent to a human. So why would we be using patient-derived xenografts? Why should we instead consider developing lab models that replicate the disease states? For example, the 3D cell culture models that I described earlier might have been a better approach. Now, as I understand it, and I'm sure people will correct me if I'm wrong, this mouse PDX models were developed across, I think, 6 or 7 centres in the US. But as far as I am aware, the NCI 60 cell line screen is still used because, of course, the PDX models are very difficult. They can be difficult to develop, particularly for ER positive, which is the most common type of breast cancer. But they're also very expensive to maintain, and the results may not always be equivocal with what you would see in a patient tissue. So my approach has been to think, well, can we increase the complexity of the models that are available? I've told you about the very simple two-dimensional models. We can make these three-dimensional. We can make these a bit more complex by adding in calves. But what about taking a tissue sample from a breast tumour, as shown here, and either using this in its entirety as an organoid, or perhaps as a little tissue slice model? So a few years ago now, we were interested in looking to see how practical using tissue slice models could be. And this was a paper that we published. And we took a primary breast tumour. We had the consent of the patient to use it in research. And a portion of it went off for routine diagnosis. A portion of it went off for tissue banking, and what was left over was embedded in a substrate, and we used 3% agarose. We then used what's called a vibratome to slice 250 micron sections, serial sections of the tumour. So effectively, we're taking these sections of the tumour and maintaining the tissue architecture. We were then able to put these into culture and culture them in vitro for up to anything between 14 and 21 days. And we were able to show that we could do viability assays, we could do proliferation. In the ones that were ER positive, we looked at the effect of tamoxifen and showed that the ER positive cell tissue slices which responded to tamoxifen were growth inhibited. And we also looked at the architecture, and we show this to our pathologists. And this is a section of normal breast tissue, which you often get when a tumour is resected. And these are two sections which show the tumour. And while perhaps the morphology wasn't top quality, the pathologist could still tell that these tissue sections maintain features which could be seen in breast tumours. And shortly after this, there was some work done by a European consortium, and they were looking to really push the tissue slice models. And their data was published in scientific reports, I believe it was. And they were able to do a whole host of different studies using the tissue slice models in 2D, as floating models, as matrix embedded models, as slices, and also as in bioreactors. And we're able to show that the tissue slice models actually were quite good at predicting how a patient might respond to treatments. The one downside that we found about using these types of models is that you're always, you're never quite sure what area of tissue you're going to get until you actually go through the actual slicing culture, and you don't know what you've got until you look at the cells at the end of the experiment. And what we were finding is that sometimes, as you would expect with breast tumours, you maybe get areas of fat, you'd get areas of normal tissue, and you weren't always hitting the area of tumour. So that's perhaps a caveat to be aware of when looking at the tissue slice models. Now, this is not my work, but it's really beautiful work that I'm sure many of you that work in breast cancer may well be aware of. This is work from Hans Clever's lab at the Hubrecht Institute in the Netherlands, and this was published in Cell in 2018. And they were able to develop this living biobank of breast cancer organoids. And what they did was to take samples from patients with breast cancer and take these little small fragments and develop these organoid cultures. Now, that was no mean feat, because that's something that people have been trying to do for, maybe not quite decades, but at least, a decade. But what the Clevers group showed that there were certain essentials shown in the dark text or non-essential components that you had to add to the cell culture medium. It's a very, very complex cell culture medium that you need to be able to successfully grow these organoids. And this is what they look like. They have this characteristic spheroid structure, and these are organoids from different tumours. And you can see that the phenotype seems to vary across different organoids, as you might expect, given the heterogeneity of breast cancer. And this group have also looked at the histology of the tissue from which the organoid was developed. So here's the tissue on the left and the organoid on the right. And this is from a ductal carcinoma or a lobular carcinoma. And this is the H&E histology, and then stained for the 3 biomarkers that are used for breast cancer diagnosis, so ER, PR, and HER2. And you can see that, importantly, all of these organoid models maintained the same phenotype, they maintained the same biomarker profiles as the tissue from which they were derived. So real conclusive proof that these organoid models are equivalent to what you would see in a breast tumour. And they also were able to identify mutant signatures. So, for example, BRCA1 and BRCA2 mutations, and the presence of BRC mutations are good indicators of the likelihood of response to PARP inhibitors. And they were able to show, they identified the different signatures, either cells that were likely to be, or tumours that were likely to be sensitive or resistant. And then using 2 different PARP inhibitors, so olaparib or niraparib, they were able to show that the PDOs also showed the same response in terms of sensitivity. In this case, they were sensitive to olaparib, but not to the niraparib PARP inhibitors. So the potential that these organoids could be used in drug screens, and they could be used to inform personalised therapy in future. And it's not just in the breast world that people have looked at establishing patient-derived organoids. And this is some work from a former colleague who used to work alongside me at the University of Aberdeen, who is interested in the gut. And they were able to take colonic mucosa and isolate the crypts from the colon. You can see here the crypt isolation. And when they were put into 3D culture, you could see the development of these human epithelial organoids, which had the epithelial cells around the outside and a central lumen, which is very reminiscent with what you see in the gut. And so these PDO models really are starting to reflect the genotype and phenotype of the original tissue. You're preserving the cellular heterogeneity and also, importantly, the structural architecture, which is associated with the primary tissue. And these intestinal PDOs have also been dissociated. So you take the tumour tissue, you dissociate them into single cells, and then put them into 3D culture. And rather remarkably, these cells seem to know where they should be in relation to their neighbours, and they form these functional organoid units. And these can be used in genomic profiling or in drug screening assays. So very sophisticated techniques now available to study or to use in the study of not just breast cancer, but also GI cancers. So what I'd like to talk about now is some of the work that we've been doing over the last 5 years or so, using this platform called Perfusion Pal. And this is the Perfusion Pal platform here. And it is essentially a pump which is attached to a cell culture plate. And within the cell culture plate, there are these wells. And within each well, we have a piece, it's a little disc of a material called Seed Easy. And this is what the Seed Easy looks like here. It's 1 millimeter in diameter, in thickness rather, 1 millimeter in thickness and 10 millimeters in diameter. And the Seed Easy is comprised of a number of randomly orientated glass fibers, and the cells sit within the glass fibers and can be maintained in 3D. And what happens is that using this artificial blood substitute, you can have an infusion of the cells. So the cells are permanently bathed or infused with this blood substitute. So the media is constantly flowing between and over the cells in culture. So the scaffold is a CDZ, and the Perfusion Pal system is it provides this, if you like, blood-like oxygen delivery. So the cells are continually bathed in oxygen. So this is what it looks like when you grow the cells on CDZ. So these are increasing numbers of cells along the top. So these are HER2 positive breast cancer cell lines. And this is the top of the CDZ and the bottom of the CDZ. So the cells grow sort of across and between the glass fiber membranes. But interestingly, under perfused conditions, this is the grey lines, compared to the static conditions, the cells are a lot more metabolically active, and this was true at all the different cell densities that we looked at. And we wanted to see if that was also reflected in the response to therapy. So in the bottom panel here, we used a drug which is commonly used to treat HER2 positive breast cancer. This was trastuzumab. And you can see that the effect of trastuzumab was enhanced under the perfused versus the static conditions. And we didn't see that in HER2 negative cell lines when we looked at these in parallel. So this Perfusion Pal system is really quite a nice way of using human-derived cell lines to study the effects of drugs. And while the slide that I'm showing at the moment was using cell lines, we have also gone on to use this more recently in patient-derived samples. So these are 4 cases of breast cancer. They're all ER-positive breast cancer. And we treated these, we applied these to the Perfusion Pal platform and treated them with tamoxifen, either under static or under perfused conditions. And what you can see is that in the dark cross-hatched bar, in 3 out of the 4 cases, we're seeing an improved response to tamoxifen in the presence of perfusion compared to static conditions. So quite a nice platform to use not only for cell line studies, but it can also be used for patient-derived tissues. And these were explant tissues that we used, and we got these from the Breast Cancer Now tissue bank. We've also been looking at developing in vitro models of normal human breast tissue, because I guess in order to really understand what's happening in cancer, we need to understand what goes wrong in normal tissues. And we like to think of these models as tools to study cancer initiation. Now, unfortunately, I'm not able to show you everything here. I had a bit of a disaster with one of my external hard drives, which has a lot of the information on this. But essentially, what we did was to take the 3 main cell components that make up the breast, the mammary epithelial cell duct, so epithelial cells, fibroblasts, and myoepithelial cells. And as I told you in the slide relating to the gastrointestinal PDOs, whereby they took the cells, they disaggregated the cells and put them back together, the cells seemed to know where they should be in relation to their neighbours. And in fact, this is exactly the same here. These are the 3D models on the left-hand side, and this is the architecture of the human tissue, the normal human mammary tissue on the right-hand side. And you can see that with each of these biomarkers, we see very similar profiles when we develop the 3D in vitro model. And we've gone on to use these models to study the initiation of ductal carcinoma in situ. And these models are very reminiscent of what DCIS looks like in human tissues. So what about up-and-coming technologies? Well, I'm sure you've all heard of microfluidics, or so-called organ-on-a-chip models. And it's even made the press. The Guardian is a newspaper which is published in the UK, and there was a piece in the Guardian about these microfluidic models. And they've been described by the Inger lab. So Don Inger, I guess, has really been driving the development of these human organs on a chip models. And what this involves is a small chip, which is about the size of a microscope slide, and it has these various channels, and you can add different cell types or tissue types to these channels, which are interconnected, and the cells can communicate with each other. And the holy grail of this, if you like, is to develop a human on a chip, whereby you can have different tissue types, so liver, guts, lung, heart, kidney, etc., bone marrow as well, to have an immune response. And the idea is that eventually these should perhaps do away with the need for using animal models in drug discovery. And there's been some quite big advances in using these organs on a chip, and some of the commercial companies now supply different organs on a chip for biomedical research. So we've also been using our own versions of these, and we've been using this to model tumor angiogenesis. So tumor angiogenesis is one of the hallmarks of cancer, and one of the original hallmarks of cancer, because, as I'm sure you all know, without developing a blood supply, or the ability to develop a blood supply, a tumor won't grow beyond a critical size of a few millimeters. So we started doing this, first of all, in 2D. So this was taking HUVEC cells, the human umbilical vein endothelial cells, which are shown here. And we seeded these onto a carpet of fibroblasts. You can't really see the fibroblasts there in the background. And depending on the type of fibroblasts, depending on which type of breast cancer they came from, we see differences in either the length of the branches. Here we can see some slender vessels forming with very little branching, whereas with this particular carcinoma-associated fibroblasts, the vessels are a lot more shorter, they're more stubby, and we're seeing quite a lot of branch points. And we've done various measurements on these to show the differences in both the branch length and the number of branches across different fibroblast subsets. But we wanted to see if we could use these models in our organ-on-a-chip models. So we have a microfluidics facility here, and we can make chips to address various biological questions. So this was looking at the 3D microvasculature on a chip. So here we have our HUVEC cells. This is the channels of the chip here. It's been sort of turned around 90 degrees in this one. So here we have the HUVEC cells in the presence of the different sets of carcinoma-associated fibroblasts. And similar to what we saw in our 2D models, the branch length differs in the 3D models, and the number of branches shown here. So this is the length of the branches, and the number of branch points differs according to the carcinoma-associated fibroblasts that we add into the model. And we've also now started to add in MCF7 breast cancer cells into these models in a way to make these more complicated. And again, what you can see is that over time, and it's particularly obvious at day 4, that you're starting to see longer branch lengths here in the CAF1s compared to the CAF2s. It is maintained over time, but it's still work in progress really to optimize the culture conditions for this, but it's quite a promising start. So at the beginning of the webinar, the Breast Cancer Now Tissue Bank was mentioned, and the Breast Cancer Now Tissue Bank is open to anybody, anywhere in the world. If you need to have human tissues, cells, serum, plasma, tissue microarrays, anything that you need to make your work a bit more humanized. So please have a look at the Breast Cancer Now Tissue Bank if you would like to consider using human tissues or cells for your research, and we'll help you as much as we can to give you the correct type of cells for your particular models. So I'd like now just to switch gear a little bit. Up till now, I've talked all about using cells and using human tissues to develop models to study breast cancer. What I'd like to spend the next few minutes talking about are so-called virtual tissue models. And this is 3D reconstructions of human tissue using virtual slides. So the slide that I showed you at the very beginning of this webinar was a section of human tissue. It was a digital whole slide image that had been scanned in. And increasingly nowadays, we're able to do that. So here we have a section of tissue showing an area of ductal carcinoma in situ, or DCIS. And what we're able to do with this is to take multiple serial sections of this DCIS, and using a process called segmentation, we can identify the areas that we want to study. And then we can use a computer technique called registration and color correction, whereby all of these sections, these 2D sections, are stacked up together, and you make this 3D model. And I think if you look at it, it really does give you a sense of depth and what's going on within the tissue. So we're not just looking at a single section, but you can see sort of all the way through. And we can artificially recolor these models, which I'll show you in a moment. And we can also rotate these tissues so that we can see different points within the 3D stack. So this is an artificially coloured reconstruction of DCIS shown in the yellow, and you can see the invasive tumour around the outside. So that's where the DCIS is starting to invade, and then the luminal spaces shown in the green. But the thing that what we really were able to notice was that the lobule from which the DCIS develops, so the lobule is this top panel A here, it's got much fewer branch-like structures, it's got the lobule has much more branch-like structures, more complex than what we see in the DCIS model, which is the panel here that I'm showing. The DCIS is much larger, but it's more simple in structure. So you can use these types of 3D models to try to understand what's going on in cancer biology, and people have used it in different cancer types, so not just breast cancer, but also colorectal cancer, and in various normal models as well. We did some studies looking at it in placenta. So back to cell culture now, and I've told you about how we can humanize our cell culture models by using different types of culture media, by using different types of approaches. But cell culture medium has historically relied on adding fetal bovine serum as a growth supplement. And that really goes back several decades, because it was noticed in the early days of cell culture that if you added fetal bovine serum, it supported the growth of these cell lines. And really not much has changed since the inception of cell culture models to where we are in the present day. And that's despite the fact that there's a number of scientific and indeed ethical issues related to fetal bovine serum. I'm sure those of you that do cell culture will know that the composition of fetal bovine serum can really vary between batches, but of course we can counter that by batch testing to make sure that we're using the same batches of serum across experiments so that we get reproducibility. FBS can also contain endogenous or exogenous contaminants, and because it's bovine origin, the FBS can affect the characteristics of your cell, so it can influence how they're likely to grow and also the genes that might be expressed. But what is perhaps less well known, I'll just have a quick drink. What's perhaps less well recognised is how fetal bovine serum is obtained. So it's a byproduct of the meat industry, and pregnant cows are slaughtered, and then the reproductive tract, which contains the fetus, is removed from the carcass. And this is done in the absence of any anaesthesia. And there's some suggestion that these fetuses may experience sentinels and may feel pain during this process. The calf is then removed, cleaned and disinfected, and the blood is collected by cardio puncture. And then the blood is clotted and the serum is separated, and off it goes to be bottled, and eventually it reaches our laboratories, and we use these in our cell culture experiments without really thinking too much about how that serum has been obtained. There are alternatives to fetal bovine serum, things like fully chemically defined media, human platelet lysate, and also serum from human subjects. But these tend to be more expensive than fetal bovine serum, and as a result, people have sort of shied away from using these. Over the last 4 or 5 years, we've been looking at identifying different fetal bovine serum substitutes which may be useful to support cell culture. And this is still very much a work in progress. There's been various people that have been involved and continue to be involved in this. And as you can see, this is our, if you like, gold standard, 10% fetal bovine serum, and the cells grow very nicely. You see this typical sort of sigmoid shaped curve. And some of the replacements, the fetal bovine serum replacements, do support to a similar extent to the FBS, some a little bit less so, but some don't support the growth at all. So like I say, this is very much a work in progress, but what we'd eventually like to be moving towards is a cell culture system which is completely free of animal components. And by that, I mean substituting the FBS, removing the need for using FBS in the experiments. So to summarise, I have this evening or this afternoon shown you various different cell culture model systems with a particular focus on breast cancer. And here are some of the advantages and disadvantages of using the different systems that I have described. I've also got rodent xenografts at the end here. But I do believe that nowadays we have sufficiently sufficiently complex cell culture models that we actually don't need to be considering the xenograft models anymore. We should be working to develop the more bespoke spheroid cultures, the mammosphere cultures, the patient-derived organoids, the organ-in-a-chip organoid cultures, etc. If you'd like to read more about these different model systems, I refer you to this paper, which was published by one of my colleagues. And if you are considering implementing animal-free research in your labs, the non-animal methodology. First of all, identify the tissue samples that you want to use. I've pointed you towards the Breast Cancer Now tissue bank. There are, of course, other biobanks that you can use if you are looking at different diseases. It's a good idea to be identifying suitable animal-free alternatives to fetal bovine serum, and I've given you some suggestions as to what you might consider using. And then, of course, how are you going to actually grow the cells? Are you going to grow them in 3D? That would be more realistic to how they are in the body. And there's various ways that you can do this. And of course, you can make your models a lot more complex. which is taking you a step closer to how tumours would be within the body, and hopefully you'll get some good results if you follow these pieces of advice. So here's some some further information that you might find useful. It's very much UK based, but you can access many of these resources if you're not in the UK. So the NC3Rs promotes the reduction, replacement and refinement of animals in research, whereas FRAME, Animal Free Research UK, and ATLA are completely animal-free organisations, and we do quite a bit of work with animal-free research on different projects. In the US, there is the Human Cancer Models Initiative, HCMI, which is looking to develop patient-derived next-generation cancer models. And if you're looking for tissues, then this is one of the biobanks that you might consider using. But of course, to really replace the use of animals in science, we need the next generation. So the next generation of younger scientists that are coming through to consider using these models and to help us achieve this goal. I mentioned Goodbye Flat Biology conferences, and this is a plug for the next conference, which takes place in October this year. I believe that the registration is still open, and it's going to be covering some of the more complex three-dimensional model systems, the so-called next-gen models, that capture the communication between different cell types that you find within tumours. And also in the UK, associated with the British Association of Cancer Research, there is a tumour microenvironment meeting, which is prefaced by a 3D model workshop, which is going to be an excellent meeting that takes place in Nottingham. And here's the QR code if you're interested in finding out more about this conference. So finally, I'd like to just acknowledge all the people in my team over the last 10 years or so that have been involved in this work and indeed continue to be involved in this work. I'm very grateful to the various funding organisations who have supported this work over the year. But I use tissues a lot in my research. And of course, the most important things to acknowledge are the patients and their families, because it simply wouldn't be possible to do this type of research without being able to use the tissues that they kindly donate. So thank you very much for listening and for watching today. And I'm very happy to take any questions that you might have. Thank you.